Okay, so Assalamu alaikum and very good morning to all of you. As usual, a very hot, humid, sticky morning. So, as I hope it's better out there where you people are residing. I hope so, but I'm not too sure about it. Anyway, as you can see, we are going to start a new system today. Our last system, as far as developmental anatomy is concerned, and that is the development of the nervous system. I started off with the development of the nervous system. I know you haven't done the you haven't done neuroanatomy as yet, but you've done neurophysiology. You're almost through with neurophysiology. So you know the basic parts of human brain. So I thought there's no harm in doing the development of nervous system and getting over with it. The parts of the nervous system you already know. You have a central nervous system, you have a peripheral nervous system, and you have an autonomic nervous system. Central nervous system comprises of brain and spinal cord. Peripheral nervous system consists of cranial nerves, peripheral nerves, and peripheral ganglia. Autonomic nervous system consists of sympathetic and parasympathetic nervous system. Now, what are the different sources from which nervous system and its different parts develop? As usual, you have surface ectoderm and you have neuroectoderm. In this case, you have two main areas from which the nervous system develops. From the surface ectoderm, we have the development of the central nervous system. You have the somatic nerves and you have preganglionic autonomic nerves. Okay. From the neuroectoderm, we have the development of somatic sensory nerves, the somatic and autonomic ganglia. Okay. Then you have preganglionic autonomic nerves, you have postganglionic autonomic nerves, and you have suprarenal gland and chromaffin cells. And then I've written focal thickening of ectoderm. But you all know the developing nervous system. We've just done the development of the special sensors. Okay. On the developing surface ectoderm, in certain places, there is the epithelium, that is the surface ectoderm gets thickened. And from that thickening develops the lens, if you remember, internal ear. तो डेवलप करता है उस थिकनिंग से, जिसको आप ऑप्टिक प्लेकोड से लेंस बना था, ओटिक प्लेकोड से इनर ईयर बना था. Then you have olfactory epithelium developing from this focal thickening. Okay, so all these structures develop from the surface ectoderm, but by a thickening of the ectoderm. A very quick recap. न्यूरलेशन फर्स्ट ईयर में पढ़ी थी, याद है? First year में we did that by day 22 or 23 the neural plate develops. कैसे develop करती है? जो ये surface ectoderm की cells होते हैं ना, ये local elongation होती है, slipper shaped structure develops on the surface ectoderm, midline of this embryonic disc की midline में over here. ठीक है, elongated structure बनता है. Now to begin with, the epithelium over here is simple columnar, but then as further development of neural plate occurs, this epithelium changes to pseudo-stratified epithelium. Okay. Now, As neurulation proceeds, a small depression appears in the midline. This elongated depression is called neural groove. The margins of these grooves approach each other, and it results in the formation of this neural tube. This neural tube then separates from the surface ectoderm and develops into a separate structure. Okay. Now this neural tube, to begin with, this is how the neural tube develops. This is the neural tube, developing neural tube. अब ये जो है ना neural tube, this develops between the first 6 somites के level पे development इसकी शुरू होती है. On the inside, I told you it is lined by pseudo-stratified epithelium. इस pseudo-stratified epithelium के close at the apical margins, it has junctional complexes. उसको छोड़ दीजिए, उसकी importance बात में. Now this neural tube is open at the cranial end and at the caudal end, but otherwise it is fused, it is closed. ये जो closure of the neural tube है ना, this starts at the midline point and then goes cranially and caudally. However, it remains open at the caudal end, it remains open at the cranial end. At the open ends, these open ends are called neuropores. Cranial neuro या rostral neuropore, दोनों terms are correct, and this is the caudal neuropore. So at the open ends, that is at the neuropores, the neural tube is in direct connection with the amniotic cavity. ये amniotic cavity. Okay, if you remember correctly, this is the amniotic cavity, and the neural tube is in direct connection with the amniotic cavity. When, as development goes on, the cranial neuropore closes. Day 25 पे जाके the cranial neuropore closes, and day 27 पे जाके the caudal neuropore closes. So the cranial and caudal neuropores, they ultimately close. The cranial rostral neuropore closes at day 25, and the caudal neuropore closes at day 27. Now, you're already aware of this thing. I'm just trying to bring back your earlier memories. The neural tube and even the neural plate, the upper two-thirds develops into brain up to the 4th somite. This part develops into brain, upper two-thirds of the neural tube. Okay? The lower 1/3 develops into spinal cord. Between 4th to 6th somite, it develops into the spinal cord. First 4 somites to cranial somite, cervical somite. So 4th somite, up to 4th somite, that is the upper two-thirds of the neural tube, develops into brain, and lower 1/3 of the neural tube develops into spinal cord. Now this neural tube has this neural canal in it. When this plate closes and forms a neural tube, obviously if it's a tube, it has a longitudinal lumen inside. This lumen, or the neural canal, this fate here, it develops into the ventricles of the brain, the empty spaces inside the brain. It also develops into the central canal of the spinal cord. And then, before I go any further with the neural tube, but the cranial end of the neural tube, you dekh lijiye. This cranial end of the neural tube is dilated. इनको neural folds कहते हैं. So these neural folds, they get expanded or dilated, and ultimately they are going to develop into the brain. So far, is everything clear? Any questions, beta? Any questions? Those who are listening, जो सो के उठ रहे हैं. Fine, no questions. Thank you very much. So I guess आपको first year में किसी ने बहुत अच्छा पढ़ाया है. Because everything has come back to you very quickly. That's great. That's wonderful. The credit goes to Madam Rehana मेरे ख्याल में उन्होंने ही पढ़ाया था. Anyway, जिसने भी पढ़ाया अल्लाह उसको नेक जज़ा दे. Okay, now let's start with formally start with the अभी तक हम उसका preliminary steps कर रहे हैं. Now, as the neural tube grows further, it develops localized dilatations. Now, these dilatations are produced because in certain areas there is very rapid development, rapid proliferation, rapid growth in certain areas, slow growth in other areas. Because of that, the areas in the wall that undergo rapid growth, they develop into dilatations. जहाँ पे growth relatively slow है, they develop into constrictions. So you have 3 dilatations because of rapid growth in the wall. अब ये 3 dilatations क्या हैं? You have the prosencephalon, which is also called forebrain. You have mesencephalon, also called midbrain, and then you have rhombencephalon, which is called hindbrain. Prosencephalon, forebrain, mesencephalon, midbrain, rhombencephalon, hindbrain. ये भी एक quick recap है. अच्छा जी, now this entire neural tube, all the regions of the brain, ठीक है, they are divided into sulci, sulcus, transversely running sulcus. ठीक है? These sulcus divide this entire neural tube into small pieces. These small pieces are called neuromeres. Okay. Now these are well marked in the rhombencephalon, that is this part. Cranially, mesencephalon and prosencephalon, these neuromeres are not well developed. Now, because of the presence of these sulci, well developed नहीं है, लेकिन that doesn't mean कि exist नहीं करता, exist करते हैं. All sulci, that is longitudinal sulcus. Sulcus आपको पता है longitudinal depression होती है. So longitudinal sulcus that divides the neural tube and all the regions of the neural tube. ये जो है lines, ये सिर्फ lines of division नहीं है, ये sulci है. ठीक है? These sulci divide the neural tube into multiple regions. अब ये जो prosencephalon, this part, this is divided into a telencephalon and a diencephalon. Telencephalon cranially, diencephalon caudally. Then we have the mesencephalon. Mesencephalon divide नहीं होता, it remains as mesencephalon. The rhombencephalon divides into 2. Cranially, we have the metencephalon, ये वाला part, and caudally we have myelin cephalon. अब ये word myelin जहाँ भी आएगा ना myel, M-Y-E-L-E, this means it is something to do with the spinal cord. ठीक है? Myelin is anything to do with the spinal cord and the caudal part of the hindbrain. Myelin sheath आपकी myelogram इसका जो टेस्ट करते हैं spinal cord पे, all these structures. So I repeat once again, these transverse sulci divide the neural tube and the different regions in the neural tube into multiple segments. These segments are called neuromeres. Caudal end पे they are more marked, इसलिए neuromeres का caudal end पे we give them the name rhombomeres because in rhombencephalon में they are well developed, so we call them rhombomeres. ये yellow जो है ना spinal cord है, okay? अच्छा जी, so ये जो transversely running sulci हैं, they divide the neural tube into multiple further regions. 3 dilatations तो हमने कर दी थी, अब आगे उनकी further divisions करते हैं. So the first dilatation, the cranial most dilatation, was the frozen cephalon of the forebrain. The forebrain gets divided into 2, namely the cranially the telencephalon and then is the diencephalon. Then comes the mesencephalon of midbrain. There is no segment, no sulcus that divides the midbrain of mesencephalon, so that remains as it is. Caudalmost is the rhombencephalon. Isme we have the metencephalon, this one, and the myelencephalon, green. This is the myelencephalon, and then this is not part of the. This is a separate thing. That the yellow part. This is the spinal cord. Okay. यहाँ पे this yellow is the telencephalon, and the blue one is the diencephalon. Okay. I hope this is clear to you all. A very quick look at how things develop from where. Starting from the caudalmost end, the myelencephalon develops into medulla, caudal part of the fourth ventricle, and inferior cerebellar peduncle. Metencephalon, pons, cerebellum, middle part of the fourth ventricle, and middle cerebellar peduncle. And then the isthmus, rhombencephalic isthmus is superior medullary velum and superior cerebellar peduncle. Mesencephalon या midbrain से you have the cerebral peduncles. अब difference है, वहाँ पे cerebellar peduncles develop हो रहे थे myelencephalon से. Mesencephalon या midbrain से you have cerebellar peduncles. Cerebral peduncles आएंगे. अब ये cerebral peduncles है. Then comes the tegmentum, tectum, and aqueduct. ये हम one by one सारा कुछ discuss करेंगे. Partly आप लोगों को already पता है. The names we are familiar with, I know. And then comes the prosencephalon, the forebrain. The diencephalon develops into thalamus, metathalamus, subthalamus, epithalamus, caudal part of the hypothalamus, and caudal part of the third ventricles. The telencephalon, the most cranial part, rostral part of the hypothalamus, rostral part of the third ventricle, cerebral hemispheres, lateral ventricles, cortex में archaeocortex, paleocortex, and neocortex, all 3 parts of the cortex, and then corpus striatum. This list was taken from Gray's Anatomy. I think आपकी किताब में भी दी हुई है. But sorry. अभी तक so far what we've done is a neural tube that is a straight neural tube like this. ये neural folds हैं. This is the neural groove that ultimately closes the neuropores close. ये caudal neuropore है, ये cranial है. and this will ultimately close. Okay. Now what happens is that fourth week में this neural tube, this begins to bend. Bend इसलिए करते हैं because it is growing longer. Space कम हो गई है. So it begins to bend and begins to bend dorsally. ये देख लीजिए, in a dorsal direction. Because of this bending, it has three sharp flexures. Flexures bend को कहते हैं. ठीक है? Two flexures are concave anteriorly, and one flexure is concave posteriorly. अभी इनको देख लेते हैं. One by one, let's do the cervical flexure. Over here, ये cervical flexure. This is at the junction of the spinal cord and hindbrain, rhombencephalon. At the junction of rhombencephalon and spinal cord. This is a 90 degrees flexure. 7th week तक ये persist करता है, उसके बाद this disappears. Okay, because the head then extends and this flexure it disappears. Then there is another flexure which is, अब ये भी देख लीजिए, this is concave forwards, concave ventrally. ठीक है? अच्छा. This is another flexure. अच्छा, color coding मैं आपको बता दूं. This is white spinal cord, brown. This is myelencephalon, pink में metencephalon. That is pink or brown together they make up the midbrain, yeah, mesencephalon. Then comes this blue. This blue is the ये mesencephalon है. This is metencephalon. This is myelencephalon. ये hindbrain के पास है. Sorry if I said any differently earlier. ये blue जो है ना, this is midbrain. ये बेटा mesencephalon. Pink is metencephalon. Brown is myelencephalon. They are both parts of rhombencephalon. Cranially, this is yellow. This is diencephalon, and white is telencephalon. ठीक है? So this white here, आप जो भी इसको कह लें, form या जो भी color है, this is telencephalon, yellow diencephalon, blue mesencephalon, midbrain, ये दोनों जो हैं forebrain के parts. Then pink में metencephalon, brown is myelencephalon. They are both parts of rhombencephalon, which is the hindbrain. And finally, this white is spinal cord. So हम mesencephalic flexure की बात कर रहे थे. This develops in the region of mesencephalon. ये mesencephalon का region है. So this develops in the region of mesencephalon, and you can see this is once again directed concave anteriorly. Okay. अब ये develop इसलिए होता है because there comes a time when the head bend occurs from a straight neural tube. When a dorsal curve develops, so there comes a time when this prosencephalon, this comes to lie parallel with this structure, rhombencephalon. ये देख लीजिए, ये दोनों parallel पड़े हुए हैं. As a result, this bend is produced, which is in the region of the midbrain. ये midbrain है, इसके region में this bend is produced. ये क्यों produce हो रहा है? Because this forebrain, prosencephalon, has come to lie parallel to the hindbrain. ये metencephalon है, hindbrain part. ठीक है? And then finally you have the pontine flexure. ये pontine flexure is between the two parts of hindbrain, metencephalon and the myelencephalon. Be directed in the opposite direction. ठीक है जी? Any questions? Any questions, beta? No questions. Thank you, Faisal. I'm extremely grateful. समझ भी आ रहा है? No questions का तो मतलब ये है ना कि बेटे, क्योंकि अभी तक तो we are doing mesencephalic flexure. Okay, mesencephalic. So there's somebody who is listening. Thank you. बेटा ये देख लीजिए. This is the mesencephalon in blue. One more. Okay. Thank you. This is the region of the mesencephalon. Okay. A bend is produced in this region, which is called mesencephalic flexure. ये bend. अब ये क्यों produce हो रहा है? Because the neural tube bend कर गई. When the neural tube bends, this forebrain, also called prosencephalon, this bends forwards and comes to lie parallel to this hindbrain. ये metencephalon है. Metencephalon is the cranial part of hindbrain. Rhombencephalon the cranial part है. So the prosencephalon, the forebrain, comes to lie parallel with the rhombencephalon. ये देख लीजिए दोनों parallel पड़े हुए हैं. They are lying parallel to each other. When they are lying parallel to each other, this mesencephalon, yeah, midbrain, this pushes relatively outwards, and a bend is produced. ये देख लीजिए, ये bend देख लीजिए यहाँ पे. This blue bends because जब तक तो straight था तो ठीक था ना, but when the forebrain bends forwards, comes to lie close to the metencephalon, as a result, this bend is produced. This bend is called mesencephalic flexure. अब बात क्लियर हुई बेटा? Welcome. अच्छा जी, न्यूरल ट्यूब की बात कर लेते हैं. Let's come back to this one and start with the histogenesis of neural tube. Okay. This is the entire neural tube. Let me show you the diagram first. This is the entire neural tube. This neural tube has this lumen. This is the central canal. Okay. The lumen inside the neural tube is the central canal. This is the wall of the central canal, yes, of the neural tube. You can see the dot, the nuclei inside the wall. These are the cells lining the neural tube. And I told you these are a single layered epithelium, which is pseudo-stratified, columnar epithelium. So pseudo-stratified columnar epithelium lines the wall of the neural tube, lines the over here. Then you have this green and blue areas. They are all parts of the neural tube. This entire neural tube gets divided into 3 zones. This innermost, this is the ventricular zone. The innermost layer, which is a lining of epithelium, pseudo-stratified epithelium, is called the ventricular zone. It is lined by different types of cells, namely the stem cells, the neuroblasts, and glioblast. Three types of cells, mainly three types of cells line the neural tube. Okay? Over here you can see this basement membrane. So these cells start from the basement membrane and go as far as the lumen. And this is the site where I told you have tight junctions. So these cells that are present in between, which is pseudostratified epithelium, this is formed by stem cells, neuroblasts, and glioblasts. Okay. Now what happens is this epithelium proliferates rapidly. Okay. Mitosis, rapid mitotic figures appear over here. Because of this, the first cells to develop rapidly are the neuroblasts. Neuroblasts, premature cells of neurons. They develop in this layer, rapid proliferation, rapid growth, and then as they mature, they move out into this layer, outer layer. The layer outside the epithelium is called the intermediate layer, also called the mantle layer. So rapid proliferation of epithelium in the ventricular zone. Yeah, this epithelial layer called the ventricular zone. Ventricular zone is it is lying next to this central canal, which will later develop into a ventricle and central canal of the spinal cord. Okay? That is why it's called ventricular zone. So ये ventricular zone में rapid proliferation of neuroblasts, and when the neuroblasts they mature, they move out into the next layer, which is the mantle layer. अब ये जो neurons हैं, they that have moved out into the mantle layer. Obviously, the neurons हैं तो उसके processes हैं. Okay? So these processes they are directed in this direction. Neurons are lying over here, their processes are directed outwards. So all these processes, they make up a third layer, which is called the marginal layer because it is close to the margin, outermost layer. Okay? This layer में you just have processes, no neurons. No neurons are present in the third layer. It is just fibers, just processes of neuroblasts present in the middle layer, the intermediate layer or mantle layer. So these processes, when they undergo myelination, they form the white matter of the CNS. Of CNS में चाहे वो spinal cord हो या brain, whatever. Marginal. अब ये जो मैं आपको बता रही हूँ, this holds true for the entire neural tube. चाहे वो neural tube brain का हिस्सा हो या वो neural tube spinal cord का ही बने. This is a general histogenesis of neural tube. That is going to be true for everything. Okay. I repeat once again, the innermost layer, the lining epithelium, pseudostratified columnar, okay, extends from the basement membrane to the surface of the canal, central canal. This epithelium is made up of three types of cells: neuroblasts, oligoblasts. Okay. These neuroblasts, they proliferate rapidly in this epithelium. So now this epithelium is given the name of ventricular zone. The neuroblasts proliferate rapidly. बाकी type of cells जो हैं, oligoblasts, they are, and glioblasts, ये जो तीन types of cells हैं, they remain there. They are dividing, multiplying, but जितनी rapidly neuroblasts multiply कर रहे हैं, they don't multiply that rapidly. So these neuroblasts, when they mature, they move out of this layer and form a separate layer outside the ventricular layer. This separate layer is called an intermediate or mantle layer. Okay? These neuroblasts have processes. These processes are directed outwards. So these processes of the neuroblast, they form a third layer. This third layer is called the marginal layer because it is the margin at the margin. Okay? These processes or fibers arising from these neurons, they undergo myelination, and once they go myelination, they form the white matter. They form the white matter of the central nervous system. अच्छा जी, अब हमने कहा था कि वेंट्रिकुलर जोन में ग्लायोब्लास्ट हैं. ये जो ग्लायोब्लास्ट हैं फ्रॉम द ये जो एपिथेलियम है, न्यूरल ट्यूब है, ये इसकी लाइनिंग एपिथेलियम है. दिस इज द न्यूरल एपिथेलियम. This glioblast moves out into the mantle layer. अब इन ग्लायोब्लास्ट का फेट क्या होता है? इसी न्यूरोएपिथेलियम से अभी जो हमने न्यूरोएपिथेलियम डिस्कस की है, लाइनिंग द वेंट्रिकुलर जोन, वी आर टॉकिंग अबाउट दैट. उसमें हमने 3 टाइप्स ऑफ सेल्स बताए थे, न्यूरोब्लास्ट, ग्लायोब्लास्ट एंड ओलिगोडेंड्रोब्लास्ट. ये जो ग्लायोब्लास्ट है, now let's do the fate of the glioblasts. The glioblasts, they develop into 2 types of cells, astroblasts, oligodendroblasts, okay, which then further develop into oligodendrocytes, okay, and the astroblast develop into 2 types of astroblasts, protoplasmic astroblasts and fibrillar astroblasts. ये आप already histology में पढ़ चुके हैं. Then there's the third types of cells. Glioblasts के अलावा इस epithelium में you also have microglial cells. These microglial cells present in this epithelium is actually mesenchymal in origin. It arises from the bone marrow. But they come into the nervous system a little later. Okay. They come late because when the nervous system develops and when this developing nervous system is invaded by blood vessels, when the blood vessels enter the nervous system, once it forms, when the blood vessels enter the nervous system, they bring with them these cells which are microglia. These microglia, आपको पता है, आपने physiology भी पढ़ी है, histology भी पढ़ी है. These microglia are simply part of your mononuclear phagocytic system. Yeah. So अब हमने different types of cells इसमें जो important cells थे वो सब हमने कर लिए हैं. Once all the cells develop and they mature, these cells, epithelial cells that make up the ventricular zone, these cells then simply develop into ependymal cells. और अगर आपको याद हो, आपने histology में ये किया था, ependymal cells are simply cells that line the ventricles of the brain and the central canal of the spinal cord. So once the ventricular zone, that is this part, once the epithelium, when all the required neuroblasts and other cells present in proliferating in this layer, when all of them are formed, then, and no more are required, then this epithelium simply stops producing all these cells and now develops into ependymal cells. Ependymal cells, आपको पता है, cells that line the ventricles of the brain and central canal of spinal cord. ये चीज क्लियर हुई? ओके, ठीक है, अगर क्लियर है तो दैट्स गुड, दैट्स वेरी गुड। अगर आपको ये समझ आ रही है। माइक्रोग्लाया रिपीट कर देती हूँ। थैंक यू गौरव। देखो बेटा, ये वेंट्रिकुलर जोन है, दैट वाज लाइन बाय एपिथेलियम। इस वेंट्रिकुलर जोन में इट वाज लाइन बाय स्यूडोस्ट्रैटिफाइड एपिथेलियम। ये देख लीजिए, ये इनलार्ज्ड वेंट्रिकुलर जोन था, ठीक है। दिस एपिथेलियम कंसिस्ट्स ऑफ मल्टीपल सेल्स में ग्लायोब्लास्ट थे, इसमें न्यूरोब्लास्ट थे, माइक्रोग्लाया भी थे, ऑल दीज सेल्स। अब हमने बात करनी है माइक्रोग्लाया की और इसके वेंट्रिकुलर जोन का फेट। माइक्रोग्लाया मैंने आपसे कहा था कि they basically are nothing much. Origin इनका mesenchymal है, आते ये bone marrow से है। Now, how do the cells of the bone marrow reach the nervous system? बेटा, this nervous system, once it is developing, once it begins to mature, blood vessels enter the nervous system. ठीक है, blood vessels में cells होते हैं, blood cells. One of them is microglia. Embryonic life में ये जो bone marrow के cells हैं, they are brought into the nervous system by the blood vessels. ठीक है, so these cells only come to the nervous system after the blood vessels have developed. Earlier development में ये नहीं होते। Earlier development में सिर्फ ये आपके oligodendroblast होते हैं and neuroblasts होते हैं। Microglia come later on once the blood vessels develop. ठीक है, और आपको इसका पता है कि it is the simply a part of the phagocytic system. And then once the ventricular zone जो है, once it stops producing all the cells that it is destined to produce, then this ventricular zone simply develops into ependymal cells. Ependymal cells की lining जो है, that is lining of the ventricles, and once ependymal cells are lined, replace these epithelium, that simply means one thing: no more cells are going to be produced. Yes. Finished. जितने न्यूरोब्लास्ट बनने थे, जितने ग्लायोब्लास्ट बनने थे, वो बन गए। Do I make myself clear? Okay. ठीक है। अब हमने ग्लायोब्लास्ट का तो फेट कर लिया था। ग्लायोब्लास्ट हम कहते हैं एस्ट्रोब्लास्ट और ओलिगोडेंड्रोब्लास्ट में develops into two types of cells: oligoblast and astroblast. Astroblast आगे से दो किस्म के होते हैं, एस्ट्रोसाइट्स में डेवलप करते हैं। एस्ट्रोसाइट्स दो किस्म के हैं, प्रोटोप्लाज्मिक एस्ट्रोसाइट्स and fibrillar astrocyte. This diagram is taken from your book. And then the oligodendroblasts, they develop into oligodendrocytes. अब further development, neuroblast. Neuroblast के साथ क्या होता है? ये तो हमने ग्लायोब्लास्ट किया ना, न्यूरोब्लास्ट के साथ क्या होता है? अब न्यूरोब्लास्ट जो है, एक बात पहले बताती चलो, ये न्यूरल ट्यूब है ना, दिस इज द न्यूरल ट्यूब, सेंट्रल कैनाल है ये, ठीक है। ये लैटरल वॉल्स हैं इसकी, ये इसका रूफ प्लेट है, ये इसका फ्लोर प्लेट है, ठीक है। तो आप ये देख लीजिए कि ये जो न्यूरॉन्स हैं, इनकी distribution along the sides is not the same. यहाँ पे देख लीजिए, it is evenly distributed. The neurons evenly distributed हैं, but as further development happens, these neurons come to lie along the lateral walls of the neural tube. Neural tube है, ये इसकी lining epithelium है, condymal epithelium बन गई है, and these neurons, they come to lie along the lateral walls of the neural tube. Roof plate or floor plate के level पे there are no neurons. ये देख लीजिए, कोई neurons यहाँ पे नहीं है, ठीक है? This region called roof plate and floor plate is simply for fibers, different fibers. ये जो neurons के fibers जो हैं, इनके processes जो है, running from one side to the other. This is the root, floor plate or roof plate. यहाँ पे neurons नहीं है, ठीक है? So अब ये neurons जो हैं, ये lateral wall में, along the lateral wall ये accumulate होते हैं. What happens to them? To begin with, ये एक large rounded cell है. It does not have a pole. It is called apolar. ये by the way Langman की drawing है, या शायद KLM की है, मुझे याद नहीं, but this is definitely KLM. This drawing बहुत अच्छी drawing है. Then ये जो apolar cell है, then it develops processes one on either side. It becomes bipolar. अब दूसरे सूरत में either one process is withdrawn या फिर ये दो processes जो हैं they fuse with each other. Once again they become unipolar. And later on they, अब कुछ cells unipolar रह जाते हैं, कुछ bipolar रह जाते हैं, but majority of the cells they develop the processes at the other end. So you have one main processes which is the axon, and then you have multiple processes which are the dendrites. Hence, this becomes a multipolar neuron. मैं इसको फिर repeat कर देता हूँ. बच्चे, neurons are produced in this neuroblasts जो हैं, in this epithelium, ventricular zone of the neural tube. These neuroblasts, when they mature, they move out into the mantle layer. अब इनकी further development, neuroblasts जो हैं, neurons that migrate out into the mantle layer, to begin with, it is a rounded structure. It is an apolar neuron. Okay. This apolar neuron then develops processes at two ends and develops into a bipolar cells. अब कुछ cells bipolar रहेंगे, कुछ की further development होती है. One process of the bipolar cell is withdrawn and it becomes a unipolar. या फिर ये कि ये दोनों processes fuse कर जाते हैं. ये आगे जाकर मैं दोबारा बता दूंगी कैसे fuse करते हैं. But this bipolar becomes a unipolar neuron, and this unipolar, majority of these unipolar cells, they develop multiple processes, so that one large process behaves as the axon, the other multiple small processes become dendrites, and this develops into a multipolar neuron. Majority जो हैं वो multipolar neuron हैं. अब बात क्लियर हुई है बेटा? Do I make myself clear? Any questions? जी बेटा. Thank you. Thank you, God. Thank you for listening to me. Okay. इसमें से nervous system में से spinal cord की development को omit ना करना, that is extremely important. Spinal cord है, pituitary है, cerebellum है, ये कुछ एक topics हैं इसमें जो बहुत important है. में ज्यादा टाइम स्पेंड करूंगी, ज्यादा एनर्जी भी इस पे मैं ज्यादा करूंगी। ओके जी, सो अब ये देख लीजिए, लैटरल वॉल ये सेंट्रल कैनाल है, द लाइनिंग एपिथेलियम एपिडेंडाइमा। लैटरल वॉल्स में न्यूरॉन्स, जबकि रूफ प्लेट और फ्लोर प्लेट no neurons, only fibers that are arise from the neurons in these plates. These fibers move from here to here, yahan se fibers idhar aaye, they pass through the roof plate and floor plate. Up as the neurons, more neurons are added to the lateral walls, ye jo lateral walls ke jo groups ye hai developing neurons के bundle, ये groups जो बन गए हैं, they simply bulge into the central canal. So these neurons, as they increase in number, they simply bulge into the central canal. As a result, ये जो lumen या जो cavity of the central canal या lumen जो है, this becomes narrow. अब what happens is, ये जो neurons पहले randomly distributed थे, but then the neurons with similar functions accumulate at one point. And उसका मतलब ये है कि the neurons with motor activity, motor neurons जो हैं, they develop in one place, they accumulate, they get together in one place. And neurons with sensory activity, they accumulate in one place. आपस में एक जिससे दोस्त मिलके बैठ जाते हैं, ठीक है, आपको पता है मल्टीपल टाइप्स ऑफ न्यूरॉन्स है, मोटर न्यूरॉन्स है, सेंसरी न्यूरॉन्स है, व्हाटएवर, मेन मोटर और सेंसरी है। सो मोटर न्यूरॉन्स ये मोटर न्यूरॉन्स है, एंड दीज आर द सेंसरी न्यूरॉन्स। सो नाउ दे एक्सचेंज प्लेसेस, अपने सही जगह पे आ गए। व्हेन दे form two main groups, the motor and sensory. There is a line of division in the form of a sulcus, a longitudinally running sulcus. इसको sulcus limited कहते हैं। ये ठीक है, ये सारा, this is simply a longitudinal sulcus running along the length of the spinal cord and brain and part of the brain as well. That limitance का मतलब ये कि ये लिमिट कर रहा है, लाइन ऑफ डिमार्केशन है between sensory and motor neurons. अब ये जो longitudinal sulcus है, इसने इस पूरे एरिया को दो में डिवाइड कर दिया। ये दो जो है, इनको have separate names, since now they have separate types of neurons. Sensory on one end, this is sensory, this is motor. Now this is the fourth floor plate, this is ventral area, this is the roof plate. Because I hope you appreciate one thing, ये जो आपका embryo है, embryo साहब है, वो इस तरह लेटे हुए हैं। That is why we have a roof plate and a floor plate. So the bundles of neurons that are divided by the sulcus limitans, you have a small, a large, this is a larger ventral and lateral, इसलिए क्योंकि it is lying lateral to the central canal. So ventrolateral bundle, the ventrolateral column, इसको basal plate कहते हैं. So ventrolateral column running longitudinally, column इसलिए कह रहे हैं बेटा, it is running longitudinally along the length of the spinal cord. So ये basal plate है. Posteriorly and laterally you have the alar plate. एक बात मैं अभी सही बता दूं, ये जो basal plate है, वेंट्रल एंड लैटरल बेसल प्लेट, इसमें मोटर न्यूरॉन्स होते हैं, और ये जो एलर प्लेट होती है which is posterior, lateral and posterior, या dorsal and lateral, जो smaller, ये sensory neurons होते हैं. Is this clear, beta? Do I make myself clear? Thank you, Faisal. आपको समझ आ गई? Thank you. अमन को भी आ गई. Good. I am happy because ये थोड़ा सा tricky है. Thank you, Star. Thank you. All the rest. अच्छा, थोड़ा सा ये tricky है. अच्छा जी, अब हम बात कर रहे थे दोनों हॉर्न्स की. one last quick last 2 minutes. आपको पता है कि इस न्यूरल ट्यूब की लोअर 1/3 डेवलप्स इनटू स्पाइनल कॉर्ड. ठीक है, अब उसी तरह 3 लेयर्स हैं. यू हैव अ मेंटल लेयर, यू हैव अ मार्जिनल लेयर. मेंटल लेयर इंटरमीडिएट लेयर होती है, और उसके बाद एक मार्जिनल लेयर होती है. मेंटल लेयर में यू हैव न्यूरॉन्स. ठीक है, सो यू हैव एन एलर प्लेट. उन न्यूरॉन्स की आपको हमने डिवीजन बता दी थी. वी हैव एन एलर प्लेट, वी हैव अ बेसल प्लेट. ये जो एलर प्लेट है, दिस विल लेटर डेवलप इनटू डॉर्सल हॉर्न. स्पाइनल कॉर्ड का कट सेक्शन आपने कर लिया है. You have 2 horns, dorsal and ventral. This is the large ventral horn. This is the large ventral horn. अब T1 से लेके L2 तक, you also have a third horn. ये lateral horn. ठीक है, ये S234 में भी होता है, उसमें भी है, सवाई के टाइप्स ऑफ सेल्स डिफरेंट है. बिकॉज S234 में आपको पता है पैरासिम्पैथेटिक न्यूरॉन्स होते हैं, जबकि लैटरल हॉर्न जो है T1 से L2, ये वाला लैटरल हॉर्न जो है, इसमें T1 से L2 के लेवल पे सिंपैथेटिक न्यूरॉन्स है, जबकि S234 के लेवल पे जो लैटरल हॉर्न प्रोड्यूस हो रहा है, इसमें पैरासिम्पैथेटिक फाइबर्स होते हैं. बीच में L3 से लेके S1 तक कोई लैटरल हॉर्न नहीं होता. T1 से ऊपर भी there is no lateral horn. ये चीज क्लियर हुई बेटा? Any questions? उरूज थैंक यू, इसरार थैंक यू. आप लोगों को समझ आ गई थी. तबरका भी थैंक यू. रिपीट करते हैं. रिपीट क्या कर दूं फैसल? यू वांट मी टू रिपीट द हॉर्न्स? हॉर्न्स रिपीट कर दूं? अच्छा, रिपीट दिस स्लाइड. ओके फाइन, ओके फाइन. देखो बेटा, इफ यू रिमेंबर योर जनरल एनाटमी, इफ यू रिमेंबर योर एनाटमी एनीवेयर, तो आपने कहा, हमने कहा था कि T1 से L2 लेवल ऑफ वर्टिब्रेट, वहां पे you have a lateral horn, intermediate lateral horn. This lateral horn was concerned with sympathetic supply, and हमने ये भी कहा था S234 के लेवल पे we have once again have a lateral horn, लेकिन S234 के लेवल पे जो lateral horn था, उसमें parasympathetic cells थे. ठीक है. Now ये वही न्यूरल ट्यूब है. दिस इज द न्यूरल कैनाल, सेंट्रल कैनाल. अब अभी हमने बात की है कि along lateral to the central canal we have groups of neuron. They are arranged in groups. Okay, we'll do that next time. I'll do this next time. Thank you. My time is over. God bless you all. See you on Monday, inshallah, with gross anatomy. 122.